In this last video of translation, we're going to talk about one example of how translation can be regulated. And we're going to look specifically at two proteins involved in the metabolism of iron, or the transport and elimination of iron from cells, and that's ferritin and the transferrin receptor. So if we look at a cell, these two proteins are important for regulating the iron levels within a cell. If the iron level within a cell is too low, then more and more transferrin receptor is going to be made that brings in more and more iron into the cell, and that could then restore iron that had gotten too low. However, if the iron levels within the cell become too high, then we increase the production of a protein called ferritin, which binds up the excess iron and brings it to the lysosome, and then that excess iron is properly disposed of. So the regulation of these two proteins, transferrin and ferritin, occur at the translation level, not at the transcription level. So let's take a look at ferritin. Okay, so again, ferritin is the protein necessary to get rid of excess iron. And if we look at the ferritin messenger RNA, we notice an important element in the 5' untranslated region. So here is an example of where the 5' untranslated region is important, right? We know it doesn't encode protein, so what does it do? Well, in this case, within that 5' untranslated region, encoded by the ferritin gene, is a sequence here of RNA that is called the iron response element, IRE. And it forms a stem loop because there is some complementary base pairing there. Now, this IRE is recognized and bound by an RNA binding protein called an IRE binding protein. That means that it binds to this stem loop here within RNA. And so at that point, though, notice that is right upstream of the start codon, meaning when this whole structure is in place, the ribosome cannot bind, and we cannot get translation of this messenger RNA into the ferritin protein. But remember, ferritin is, so at this point, ferritin is turned off. We only need protein being made when there's too much iron. So when the iron concentration, and I'm answering this question here, when the iron concentration increases within the cell, then it's going to be more likely that iron is going to be bound to the regulatory site of this IRE binding protein. Iron is a regulatory inhibitor of this protein, so that when iron increases in concentration within the cell, it will bind to that IRE binding protein. That protein is no longer active. It can no longer bind to the IRE, and therefore the ribosome can now bind and start making protein. Now that ferritin can start soaking up some of that excess iron, delivering it to the lysosome, and thereby getting rid of it. Okay, well, what's interesting is that the regulation of transferrin receptor, so if we come back here, we're looking at the receptor here, that when iron levels are low, we want to increase the amount of transferrin receptors. Well, it's regulated by that same system. It's co-opted and used in a slightly different way. So here, again, there is our IRE, and that is encoded by the transferrin receptor gene and is therefore found in the transferrin messenger RNA. It is found in the 3' untranslated region, so it doesn't give rise to protein. Right? This is another example. Well, hey, wow, sometimes this 3' untranslated region can be quite large. Is it just junk or does it do something? Well, in this case, it does something very specific. It contains this IRE, which forms a stem loop, which is then bound by the IRE binding protein. Now, what this does here, unlike what we saw in the ferritin protein, what it does here is it protects the 3' end and therefore all of the messenger RNA from degradation by endo and exonucleases. So as long as we have the IRE binding protein binding to the IRE, we're good to go, and we have translation of this messenger RNA and the production of transferrin proteins that allow the cell to bring in more iron. Okay, so we need it when iron levels are low. Beautiful. That's the only point in time that our IRE binding protein is active. Now, once the iron levels in the cell begin to increase, the cell no longer wants to make this protein. The cell no longer wants to bring any more iron in. Perfect. As the iron levels increase, they bind that IRE binding protein. They inactivate it. It can no longer bind the IRE, and now the messenger RNA becomes vulnerable to these degradative enzymes, to these exo and endonucleases, which is perfect. As soon as the concentration of iron is too high, we no longer make transferrin receptor protein and thereby no longer bring iron into the cell. So this is a great example for a couple reasons. Number one, it tells us why 5' UTRs exist. What can be important about them? Why 3' UTRs exist? What can be important about them? And finally, it shows us that the systems of regulation don't have to be constantly refigured out. A single regulatory mechanism, this IRE binding protein binding to an IRE, can be recycled, right? Evolution can reuse components that the cell already has and just reuse them in different ways. In this case, we have an IRE in the 3' that's protecting the RNA from degradation. And in this particular case here, we have that same IRE, we have that same protein that binds to it, in this case inhibiting translation. And that's the way evolution works. It uses components that the cell already has in new and novel ways. Okay, next and lastly, we're going to discuss DNA replication and DNA repair in the final lecture.